Hello and welcome to Meditut, the medical tutorials. Our motto is understand, learn, and give your best performance. As discussed in lecture 4 of Meditut, the amphipathic lipids have a polar head and a non-polar tail. And a very important role played by the amphipathic lipids is that they act as surfactants, means they reduce the surface tension. If the amphipathic lipid is present in a water-based medium, it forms a layer on the surface. But suppose this amphipathic lipid is present in a enclosed space, means there is no surface to go. For example, the amphipathic lipids are present inside the blood vessel, in the plasma. So there is no surface to go. So how will these non-polar tails run away from water? In this case, it forms a spherical structure like this. All the non-polar tails are directed away from the water, from the surrounding water, towards the core. It forms a spherical structure where all the non-polar tails are away from water and the polar heads are on the surface towards the water. This is called micelles. So amphipathic lipids form micelles. What are micelles? Micelles are basically spherical structures formed by monolayer of amphipathic lipids in such a manner that polar heads are on the surface and non-polar tails are directed towards the core. These are the important points to remember about micelles. Number one, it is a spherical structure. Number two, it is a monolayer of amphipathic lipids. Number three, polar heads on the surface. And number four, the non-polar tail towards the core. So micelles, they have a non-polar core and a polar surface. So they can even carry a non-polar molecule within their core. So the important role played by the micelles is in the lipid transport in plasma. The lipids are transported in plasma is basically water-based medium, an aqueous medium, and lipids are basically non-polar. So how they are carried in plasma? They are carried in this form, the lipoproteins. Lipoproteins are basically the micelles, the VLDL, LDL, HDL, chylomicrons. These lipoproteins, which transport various types of lipids in plasma, are basically in the micellar form. So this is a very important physiological role played by the amphipathic lipids forming micelles. Suppose we have consumed fat, we have consumed lipid. So due to the peristaltic movement, this lipid which is present in the GI tract, it will be broken down into small chunks, small lipid droplets. But these droplets, they have a tendency to coalesce again because all of them are hydrophobic. All of them are running away from water and surrounding medium is basically water. The GI tract lumen is basically containing the water-based medium. The GI secretions are basically water-based. So these lipid chunks, these lipid droplets, they have a tendency to coalesce and form a lipid chunk again. But in the GI tract, in the GI secretions, if an amphipathic molecule is present, these amphipathic molecules, these amphipathic lipids, they form a layer around each lipid droplet in such a manner that non-polar tails are directed towards the lipid droplet and the polar heads are towards the surface. So these lipid droplets will remain suspended in the water-based medium. They will not coalesce with each other. They will remain suspended in the water-based medium, means the lipid which is normally insoluble in water is made soluble in water. This process is called emulsification. A non-polar molecule is made soluble in the polar medium. This is called emulsification, and this is a very special role played by the amphipathic lipids. They act as emulsifying agents. And which amphipathic lipids act as emulsifying agents here? The bile salts. Now, the important role played by the bile salts is that they act as emulsifying agents which helps in lipid digestion and absorption in the GI tract. Now these lipid droplets, they can be easily acted upon by the enzymes and they can be easily digested and subsequently absorbed in the bloodstream. So this is another important role played by the amphipathic lipids as emulsifying agents. Suppose there are two mediums which have to be separated and there are two aqueous mediums which have to be separated. This is one water-based medium, this is another water-based medium. Let us suppose this is cytosol and this is the ECF. The two water-based medium, they can be separated by a non-polar medium only. But this non-polar medium has to be a stable medium. This cannot be stable because this has a tendency to coalesce. So to make this non-polar medium stable so that these two polar mediums remain separated forever, this is made possible by a bilayer of amphipathic lipids. So this is called membrane. So membrane is basically a bilayer of amphipathic lipids formed in such a manner that non-polar tails are forming the body of the membrane. The polar heads are directed towards the outer side and towards it is like a sandwich. Bilayer. So polar heads are towards the outer side and towards the inner side, and the non-polar tails are forming the body of the membrane. So this is a basic structure of membrane. Number one point to remember about membrane is it is a bilayer. Second is the polar heads towards the outer side and towards the inner side, and the non-polar tails form the body of the membrane. This is different from micelles. In the micelles, number one, micelles is a monolayer and membrane is a bilayer. Second is, in the micelles, the polar heads are towards the outer side and the non-polar tails are towards the inner side. But in membranes, both sides, inner as well as outer, are the polar heads. Secondly, in the micelles, the core is non-polar, the surface is polar. But in membranes, the core is also polar, the surface is also polar. This is the basic structure of membrane, the bilayer of amphipathic lipids. All the membranes, they have the same structure. It may be plasma membrane, mitochondrial membrane, nuclear membrane, lysosomal membrane, endoplasmic reticular membrane. Any membrane has this structure, the bilayer of amphipathic lipids. Another important role played by the amphipathic lipids that they form a liposome. Well, you can see liposome is just like a membrane. The bilayer of amphipathic lipids, the core is polar, the surface is also polar, and the body is the non-polar tails. Well, membrane is a natural structure. Liposome is a synthetic structure. And why this synthetic structure is formed? This synthetic structure is synthesized. Role of the liposome is for targeted drug delivery of toxic drugs. For example, some anti-cancer drug is to be delivered to the cancerous tissue. The problem with the anti-cancer drugs is they have a systemic toxicity. To reduce the systemic toxicity, we target the anti-cancer drug to the cancerous tissue only. So this drug will be carried in the liposome. Suppose there is a hydrophilic drug. It can be easily carried in the core of the liposome. Now this is protected from the outer environment till it reaches the target tissue and delivers the drug to the target tissue. There are means how it reaches the target tissue and delivers the drug specifically to the target tissue, but till the time it is protected from the outer environment. If there is a hydrophobic drug, it can be carried in the membranous portion of the liposome because this is the non-polar portion of the liposome. Another important role played in the liposome is in the gene therapy. If a gene has to be delivered, suppose the DNA has to be delivered to the target tissue, or even RNA is to be delivered, like siRNA therapy, the small interfering RNAs. This siRNA therapy is being tried for age-related macular degeneration. So liposomes are important vehicles for transporting the DNA or RNA to the target tissue. So these are the important roles played by the liposomes. So let us see which liposomal drugs are already present in the market. Amphotericin B, the antifungal drug, doxorubicin, daunorubicin. These have already come in the market as liposomes in the liposomal form. And clinical trials are going for these drugs: vincristine, cetarabine, cisplatin, morphine, amikacin, and vaccines for hepatitis A and influenza. So these are the various uses of liposomes. Let us quickly revise the special roles played by amphipathic lipids in human body. We discussed in lecture 4 also the special role played by the amphipathic lipids, and in this lecture also. So let us quickly revise all the roles played by the amphipathic lipids in the human body. One is they act as surfactant, reduce surface tension, as we discussed in previous chapter. Secondly, they act as micelles, they form micelles which help in the lipid transport. Third is they act as emulsifying agents. Fourth is they form membranes, and fifth is they form liposomes. Surface tension surfactant role we already discussed in previous chapter, that which helps in preventing the alveolar collapse, and it prevents the respiratory distress syndrome of the newborn. And the role of amphipathic lipid as surfactant helps in diagnosing obstructive jaundice by doing Hayes sulfur test. Micelles, they help in bile salts which form the micelles. They help in digestion and absorption of lipids, and the lipoproteins which transport lipids in plasma. Emulsifying agents, bile salts, they help in digestion and absorption of lipids. And liposomes, these are the synthetic molecules useful in targeted drug delivery of toxic drugs and for gene therapy. If you wish to better your performance in any of the exams and build a solid foundation for a great career in medical science, please like, share and subscribe our channel Meditut the Medical Tutorials.